In this paper, we explore the role of nuclear oscillations in chromosome pairing during meiotic prophase in fission yeast and find that these oscillations promote the dynamics of pairing and unpairing to avoid excessive connections and missegregation. In Figure 4, we show that prolonged chromosome pairing is accompanied by missegregation. We stopped nuclear oscillations by using a microtubule poison MBC and followed the zygotes until the end of meiosis II. In panels A and B, we show two examples of cells in which the spindle pole body is marked in magenta and homologous loci in green. In the first cell, nuclear oscillations were stopped at the time when the loci were paired, whereas in the second cell, the loci were unpaired. The first cell ended up with missegregated loci. They were all found in one half of the cell at meiosis II. In the second cell, the segregation proceeded normally, with each of the four loci close to one of the four spindle pole bodies. Panel C shows the position of the loci in green and of the spindle pole body in magenta for the cells from panels A and B. We see that in the cell in which the loci were paired, all the loci segregated together with one spindle pole body at meiosis I. In the cell with unpaired loci, the segregation was normal. In panel D, we see that the distance between the loci during the MBC treatment in the cell with paired loci was indeed smaller than in the cell with unpaired loci. Panel E summarizes the quantification of segregation patterns for 5 conditions. In control cells, segregation was normal. In cells where the oscillations were stopped with either MBC or another microtubule poison TBZ, segregation problems occurred only when the loci were paired. These results suggest that prolonged chromosome pairing is accompanied by missegregation. To infer the molecular mechanism underlying the observed missegregation, we examined the segregation of the whole DNA mass at meiosis II. We observed a single DNA mass in most of the zygotes with incorrect segregation in which the oscillations were stopped when the loci were paired. On the contrary, the cells with unpaired loci had 4 DNA masses, just as untreated cells. These findings indicate that the missegregation observed in our experiments was a result of a failure in segregation in meiosis I, suggesting that prolonged pairing induced by inhibition of oscillations is accompanied by irresolvable recombination intermediates.